right. So we're gonna get started on exercise 2 because it takes the longest amount of time. This is looking at photosynthetic efficiency of different photosynthetic pigments. So what light colors they absorb, or wavelengths that they absorb. Then we're gonna do exercise 1 while exercise 2 is done. Okay, cool. So I'm going to demonstrate this really quickly. We're going to do chromatography. It takes about 30 minutes to set up and run, but we're going to have you guys create a hypothesis just after you guys start getting the ball rolling. So what you first have to do is take this chromatography paper. You guys have
Okay, so for exercise 2, you want to cut about 4 inches of the chromatography paper, like so. And then what we're going to do is we're going to cut a picket-like fence line at the bottom, like so. That's the part that's going to sit in the chromatography solution in your tube. And then we are going to draw a line with spinach juice right here, where it sits just above the chromatography solution in the tube. Okay, so now I drew my spinach juice leaf just above that picket line fence, and I'm going to put it in the chromatography solution in one of the tubes in these trees right here. And each student is making 2 strips each, and see how it sits. The line, the loading line of spinach juice sits above the chromatography solution. That is required so it doesn't just dissolve all the juice into the solution, and you can slowly see the different pigments such as chlorophyll A and chlorophyll B and xanthophyll, which is that yellow stuff. It's slowly running up the chromatography paper as the paper is absorbing the solution. So we're going to let this run for 15 minutes. Each student is going to make 2 each. And the goal of this is to separate those pigments individually so later we can look at their absorbance. So we're going to let this run for about 15 minutes while we do exercise 1.
on your desks, you also have some scissors and some rulers. And you're going to want to cut about a 4-inch strip. So it's going to look like a roll. I just do this for the camera, so bear with me and moving that in place. So you're going to take this roll and you're going to measure about 4 inches. And then what you do is you take your scissors and you want to flip it so you have a nice little arrow at the bottom of this chromatography paper. And what we're going to be doing is placing it in chromatography solution, which like slowly evaporates and allows the liquid to run off the paper and separate different pigments. So when it's finished, it should look like this when you cut it. But we're going to be looking at specifically in plant leaves. So we have some organic baby spinach for you guys today. The way that we have to run this though is that they all have to start at the same starting point. So you want about like a Sharpie's marker thickness, no more than that, of spinach juice on this piece of paper. So if you can look you can look at this diagram right here, because it's a little bit easier to see, and I will be walking around the other classrooms while showing it. But you want a really thin line. You want it to be so thin, but you want it to be thin. If you have a really fat line, it's not gonna work, and you're gonna have to make another one. So if you mess up, not a big deal, just start another one with a new piece of chromatography paper. So you're gonna have it like this, like I mentioned. And then these trees are going to be set up. There are jars of ether you're going to put them in. Make sure they evaporate very quickly. So if you do take the cork off, just make sure you put it right back on. And you're going to place your piece of spinach paper in here. Make sure your spinach line is above the liquid. You don't want it to dissolve in the liquid. You want it to run up the paper. So the important piece is that when you put it in your bit of acetone, only the tip and not the spinach is going into that solution. and you can watch the liquid run up this water, run up this piece of paper. That is actually gonna take some time. That's why we're doing this now, and we'll talk about generating the hypothesis for this a little bit later. There are pre-made out bits of acetone over there. Ask the TA for help for adjusting it and putting it on these trees. These bits, move this up and down, we don't need that. I would use these little screws right here to secure your glass jars right there. And you want to make sure that they're not so tight that you're shattering them, but that they're not going to fall out. And then remember to put that fork back on. Sound good? Awesome. I'm going to go run over there and demonstrate what it looks like for that. So go ahead. Oh yeah, and you're going to make 2 each. Awesome. So there should be scissors, rulers, chromatography paper, and front of you. 2 per person. Baby spinach is up front.
Here. I think it's 2 and 1 too. That's what they're saying.
Oh, gotcha.
And there's only, like I said, it's only 12 places per bank.
Do you remember that song? Baby shark.
Exactly.
Baby spinach. I know, it's terrible. I have to entertain myself. It's a long 10 hours for me. Starts at 8, doesn't end until 6.
Yeah. You teach them 1.
Yeah. You can let them know office hours for an hour and the next. And then my brain is just mush. Mostly I want to just disrupt things as much as possible.
It's finished. All right, we're gonna move on. All right, we're gonna go over the basics of photosynthesis, what you're gonna need for this lab today. All right, so photosynthesis, it's an energy capturing process. We talked about it, it's the opposite of cellular respiration. So, who does it? Plants, some bacteria, and some other unicellular organisms. So even some unicellular eukaryotes and photosynthesize, which is pretty cool. All right. So here are the basic components of photosynthesis. Sunlight, water, and CO2 are those inputs. They use enzymes and they use photosynthetic pigments such as chlorophyll and different kinds of chlorophyll to capture the light energy from that sunlight. So they need those pigments to capture that energy. And then they can harness it and turn it into chemical energy, which they store as sugar, C6H12O6, which is made of sugar, and a byproduct is oxygen. Everything good over there?
Yeah, he needs to go on.
Okay, yeah, we can finish this. Okay. This computer so much. All right, so I want you guys to know those inputs and outputs. Chloroplasts, in contrast, are the organelle where the process of photosynthesis occurs, especially in plants and eukaryotic algae. So all these eukaryotes that do photosynthesis have chloroplasts. You guys should know the general setup of these chloroplasts, and we're gonna look at a video really quickly that kind of shows you a bit of a breakdown for exercise 1. But with photosynthesis, we can break it down into 2 different parts. First is the photosegment. This is where light is captured and temporarily stored in energy storage molecules. And this is where water splits and forms oxygen. And this occurs specifically in the thylakoid membranes in the chloroplast, in that organelle. So the second part is the synthesis part of photosynthesis. And this is where the actual sugar or glucose is constructed to capture that chemical energy for long periods of time to use later. This occurs in the stroma of the chloroplast in that little organelle. So you can think of it as like a cell almost within a cell. So stroma is kind of just like that free open space within that organelle, within the chloroplast. So we just talked about the chloroplast as the organelle, but then there's also chlorophyll, and that is a pigment. That's the green pigment found in plants. That's what we see when we look at a plant and it bounces back in our eyes and we read it as green. It's responsible for that absorption of light to capture that energy temporarily, where then they can turn it into glucose to store for later. This is a really cool close-up photo of chloroplasts within cells. You can see them like the little green bits. And then just to clarify, chloroplasts, they're the organelle. Chlorophyll is the pigment. Those are 2 very different things and they're not interchangeable. Just an FYI, because those are really easy to muck up in the brain. Okay, so now let's just think about what sunlight is for a second. So sunlight, we see it as white light. But if you guys have taken a chemistry class or any physics classes, you know that white light is actually a combination of a bunch of different wavelengths. And the visible light spectrum, which runs from 740 nanometers to 400 nanometers in wavelength. The wavelength determines the color that we actually see, that frequency. So we perceive different frequency wavelengths of visible light as different colors. We can't see certain things outside of visible spectrum. That's where we get like microwaves and UV radiation. That's stuff that we can't see, but it actually exists. Fun fact, other organisms actually can see UV lights, or like some butterflies, they see flowers completely differently. But for us, the visible light spectrum is 740 to 400 nanometers. Okay, so just a thought experiment really quickly. We see light based off of what light wavelengths are bouncing back, right? So it's whatever wavelengths are coming into our eye after we interpret it to a certain color, right? So knowing that, if white light, which has all the rainbows of the colors, are shining out onto something, and it bounces back blue, like we look at the sky and it's blue, why do you think that is? Is it because all the blue is being absorbed, or all the other colors are being absorbed and the blue is being absorbed? The blue being, so thumbs down if blue is being absorbed, and thumbs up if blue is being reflected. Yes, blue is being reflected, which means all the other wavelengths, all the infrared or like deep red and all the other lower frequency stuff can actually escape our atmosphere, like the gas. The gases in our atmosphere make it so that everything else is absorbed, but blue is actually reflected back from what we see it. So if we think about leaves using a green photosynthetic pigment to absorb energy, are they absorbing energy from which wavelengths of light? So like red, orange, yellow, green, blue, do you think they are absorbing? So I just want you guys to chat with your neighbor about that. And generate a hypothesis. Or exercise paper.
So I would say that.
All right. So do you think that chlorophyll, that photosynthetic pigment that absorbs light, do you think it specializes in absorbing energy from green pigments or other pigments? Other pigments, cool, good, because it's reflecting that for you, right? So then you can use the rest of that for you guys to hypothesize. It doesn't have to be right. Remember, our hypothesis is just an educated guess based off your observations on so far. As to what pigment you guys think it will absorb most often. You guys can write that together in a little bit when we have a little bit more free time. I just wanted to walk you guys through that first bit of thinking. All right, so we're gonna jump back to exercise 1, and we're going to focus on doing diagrams of a leaf cell. So really quickly, I'm gonna exit out of here, and we're gonna watch a really quick YouTube video. And it's gonna explain the purpose of a lot of the different cell functions or the different tissue functions in a leaf.
So we know that plants make their own food through photosynthesis. But how does a plant get together the ingredients it needs to? Sunlight, carbon dioxide, and water, and then combine those to create glucose and oxygen? Well, that's what leaves are for. A leaf is like a plant's food factory, and its parts work together to get the reactants into one place so that photosynthesis can happen. Let's start with sunlight. Have a look at a leaf. The top of it is exposed to the most light, so the cells specialized for trapping light are on top. These cells are called palisade mesophyll, and they're packed full of chlorophyll, the green chemical that plants use to absorb light. That's why the top side of the leaf is darker than the bottom side. You should also note that most leaves have a large surface area, so they can trap as much sunlight as possible. What else do we need for photosynthesis? Carbon dioxide. That's where the bottom of the leaf comes in. It's got these little pores called stomata, which open up so carbon dioxide can diffuse into the leaf. They're controlled by sausage-shaped guard cells, which open up to let carbon dioxide in, but they can also close the stomata to prevent other things like water from escaping. So the carbon dioxide comes in through the stomata, makes its way through the gaps in an airy layer of cells called the spongy mesophyll at the bottom part of the leaf, and then heads up to the palisade layer for photosynthesis. Leaves are thin so that the carbon dioxide won't have too far to travel. So we've got sunlight and carbon dioxide now. What's left? Water. Water comes up through the roots and stem and enters the leaf through a vascular bundle, which contains a hollow tube for water transport called the xylem. Look at how the vascular bundle spreads out in the leaf to form veins so that it can spread water throughout. So now the leaf's palisade cells have water, carbon dioxide, and sunlight. All they need to photosynthesize and make glucose, their food, and oxygen. But how does the leaf prevent unwanted intruders like bacteria from getting in and stop important reactants from escaping? Above the palisade mesophyll and below the spongy mesophyll are epidermis cells, which produce a waxy coating called the cuticle. The cuticle seals up the leaf so that the only way in and out is through the stomata, which are regulated by those guard cells. Now let's go back through the parts of the leaf, from top to bottom. We've got the thin, waxy cuticle, then the epidermis cells. These basically make up the leaf's skin. Then comes the chlorophyll-rich palisade mesophyll, where the bulk of photosynthesis occurs. Below that's the spongy mesophyll, which have plenty of space in between them for reactants to move through. Through that space stretches the vascular bundle, with xylem to transport water and phloem, which transports sugars after photosynthesis. And below that, we've got another layer of epidermis and cuticle. Spaced around the lower epidermis and the cuticle are the stomata, with guard cells on either side. Leaves come in all different shapes and sizes. Look at a cabbage leaf and an oak tree leaf. But most of them have these same parts in common, which allow them to get their reactants in and perform photosynthesis.
All right, so that pretty much answered a lot of questions that you guys are about to fill in for exercise 1. All right, so exercise 1, you guys are going to be looking at leaf structures. There are prep slides on both sides of the classroom in those green little boxes. And you guys are going to want to draw and label a nice cross-section photo of these. Make sure you write all the functions down too, and that this drawing is sufficiently large so you can actually study it and understand what these are. Sound good? Okay, cool. That's pretty much exercise 1 right there. So if you guys want to go ahead and grab a microscope and a partner and the slides, we can, and you guys should be able to get an image by now, but you can help us know. If you guys are on, I have to be able to, okay. So this is like a cross section of a leaf. If you guys can see it,
Wait, you don't need that.
okay. This is see this like tightly packed clear line of cells right here and right here? That's the epidermis layer, remember? So that's like the skin of the leaf, if this is a cross section of the leaf. The stomata, they aren't actually cells, they are just the openings, the little holes where gas can diffuse in. So these little holes right here, that is what I would call a stomata. These little bunchy bits right in front of both of these holes, those would be the guard cells. So those are cells that swell shut when they feel like they're losing too much water to the environment. They shut up and they don't get as much gas to make more glucose, but it does keep them from drying out and looking like a basal leaf that has not been watered. So then what you have is the spongy mesophyll right here versus the palisade mesophyll. You can tell the difference between them because see how loosely packed these are? They're loosely packed so air and oxygen can diffuse through them all the way through to the palisade mesophyll. The palisade mesophyll is much more tightly packed and they are going to be packed full with the chloroplast because they're on the side that's closest to them. Does that help with these drawings? Okay, cool. I was like, I think everyone's talking about it. Because in real life, these cross sections, not every single part of like where you're looking is going to have very perfectly laid out bits, but they are all there, I promise you. It just depends on the cross section you're looking at. So this is a really, really pretty cross section that kind of demonstrates everything you guys need to know.
Oh, yeah. No, no, I thought.
All right, so for exercise 2, now that we have our little picket fence with all the different pigments separated because they ran for 15 minutes, we removed them from the chromatography liquid. And now what we want to do is separate chlorophyll A and chlorophyll B from the rest of the paper, because these are the pigments we want to isolate and look at their absorption. So what we're going to do is take our scissors and essentially clip only the area that looks like it has chlorophyll B on it. So we would cut all the parts of this paper that we could see clearly chlorophyll B, that darker green pigment. It doesn't matter the shape, we just want to get the paper that has that pigment in it. Then what we're going to do is cut chlorophyll A separate from the xanthophyll, so those little yellow lines, and separate from the chlorophyll B. So the parts where you can't really tell what color it is, I would just not include those and just cut the big sections where you know it's chlorophyll A. This is not that big of a deal if you miss some of it, because with your group, which is the front of your table, you actually combine all your little pieces and then you put them into a big tube of acetone, the little sections of paper that we're cutting out. For chlorophyll A, you'd have your own little tube that you have acetone in, and you're dissolving those pieces of paper. But because each of you made 2 of these strips and all of them got cut out that are good, you'll have one big tube with a bunch of little pieces with chlorophyll A pigment in it and a bunch of little pieces with the chlorophyll B pigment separated in its own acetone tube. So there should be enough that those pigments will dissolve in our acetone liquids, which we can then put into the spectrophotometer later.
So we are going to take our chromatography paper and move on with exercise 2. So if you can see this, I'm not sure if you can. So chlorophyll B should have separated from chlorophyll A on your paper. And chlorophyll B should be a little bit more like yellow green. We're gonna cut them apart. So your group mates around you, you guys are gonna essentially combine them all, and we're going to put them in a little bit of acetone, which is over here. And you're going to dissolve them into, you're gonna put all your pieces that you cut apart into either the chlorophyll A or the chlorophyll B tube you guys have. So you're going to have chlorophyll A and chlorophyll B tubes. You cut those little pieces apart, and you really don't want to get the stuff where there's both of them in there. So if there's a little bit of overlap, just ignore that part. Just cut the parts that are really clear, either really, really blue or really, really like yellow green versus blue green. And then you're gonna drop all those little pieces in there in your group, and then you're gonna put acetone over them so it's gonna dissolve that stuff into this fluid. And then we're gonna talk a little bit more about how spectrophotometry works. Yes? Okay, yes, do that. If you guys need help identifying what the blue versus the dark green is, ask the TA.
So it can be below that yellow one. The yellow is really just.
So I just really want to quickly go over what we're doing with spectrophotometry. So what we're doing with spectrophotometry is measuring the actual absorbance of those photosynthetic ions you guys now have dissolved in your tubes. So I'm gonna hold this up for you guys too. What you can see here is like it's a light green liquid. See how that pigment dissolved into that acetone right here? We know acetone is very clear. If you shoot white light to it, it's not going to change the absorbance unless there's something in that acetone. So we have these really, really cool devices called spectrophotometers. And what they do is they shoot white light through a prism and then through our sample solution. And what it does is it can help us measure what wavelengths are being absorbed and what wavelengths are being passed through our sample. And our sample is our solution with the chlorophyll in it. And so how this works, just really, really simply, is that it has this really cool wavelength selector. So what it does is it takes the white light, it shoots it at a prism, which we know separates those all out so you get the really cool rainbow. And then what happens is that there's a little slit and it adjusts so it only lets a specific wavelength. So it's like we're only gonna look at red light. And then it shoots red light through our sample solution. And based off of a blank run where it runs through clear liquid, it knows how much to go through if nothing is being absorbed. But then it shoots it through our sample solution, and then it can tell, wow, it normally has this many like photometers, and it's showing it this red, but now it's much faded, it's much more faded. So you can, that's what it's trying to demonstrate here, is it's really bright yellow here and a little more faded because some of that yellow was absorbed by our solution. Which essentially, eventually tells us how much is absorbed of yellow wavelengths. What these specific spectrophotometers do is it does that, but it does it at a bunch of different wavelengths. So it actually tests, here we go, a bunch of different wavelengths, all the way throughout the spectrum of visible light. So that little slider goes through and tests all the different wavelengths, and it can give us an absorbance spectrum of our solution. So it can tell us, oh, it absorbs this color really, really well, but it doesn't absorb yellow very much at all. And so that is what we're trying to go through with our spectrophotometer, looking at the absorbance specifically of chlorophyll A and specifically of chlorophyll B, and see if it's actually reflecting that green light or it might be absorbing the blue light. Remember, our hypothesis is that it's actually reflecting green light because that's what we're seeing. We're seeing that green come off of it. So if green was right here in the magnetic spectrum, we would expect the absorbance to be high or low. Yes, because it's reflecting off of that part. Cool. So that's pretty much what we're doing with the spectrophotometer. And that is why we chopped up those little pieces and we're putting them into solution, because we're going to be shooting them through the spectrophotometer. And we're going to get one of those graphs I just showed you. So, okay, so what you can see is a spectrophotometer right here. And this little hole right here is where you can put your specimen. So you guys have chlorophyll A, chlorophyll B solutions with your little pieces of paper. The little pieces of paper would obviously mess with the absorption. So that's an issue. So what we can do is we can take these and we can pour just the liquid with the dissolved solution into our handily labeled A and B chlorophyll test tubes, which can then fit into our little SpectroViz thing right here. Right? Okay, so when you turn it on, it's going to have you press through a couple of buttons, and then it's going to ask you to run a blank solution. It's going to like tell you to insert a blank. That's essentially giving you the baseline, and you guys are supplied a blank. It should say blank on it, and it's just pure acetone. We know pure acetone will absorb nothing because it's perfectly clear. It'll just shoot that white light through it. It's a good baseline that it can then prepare all other different colored liquids to. So then we can put that in here and we click the run button on the spectrophotometer. Once it starts running, it'll tell you when it's done. And then you are ready to actually measure your own chlorophyll A and chlorophyll B absorbance spectrums. So then you put them in here and you click run. Yeah, I don't know if you guys can see this. Oh, there we go. You click run, and then it'll show this cool rainbow graph. If it's not up, come find one of the TAs, they'll help you bring it up. And you will see a graph of actual absorbance, so like a little peak and trough in the different spectrums for your sample. You can take a photo of that, and then that is what you're going to be drawing. A line for chlorophyll A, and then a line for chlorophyll B on that graph you guys see your line. So I want you guys to copy that out. Does that make sense? Okay, so you might have to like look through a couple of prompts, but you will get to it where it's finding your point.
I mean, it does go up. Just know that this is exactly, yeah, this is like
Okay, so they should look something like this. Let me just make sure I'm still sharing my screen on Zoom. Share screen. Okay, so they should look something like this. You guys had a big rainbow background, but this is just demonstrating. We can see roughly, like I said, 300 to 740 whatever nanometers. That's visible light spectrum, with the bigger ones being the red end of the rainbow, smaller ones being blue. And chlorophyll A and chlorophyll B should have their biggest peak in the ultraviolet area, like approaching ultraviolet area, so like in the blues and the purples. That is the wavelengths that they are the most efficient at absorbing from, which means that they can get a lot of energy, fix a lot of energy from those colored wavelengths. And then also, there's another peak right here in the deep red area, but then there's a trough in the green. So since we talked about the hypothesis being that since they reflect green light, or that they reflect green light, does this graph show that? Yes, because if this is 100% absorbance, if you're labeling this 100% absorbance and this is 0 absorbance, it's clearly reflecting all of the green light coming at it. So you can use this graph and that interpretation to see whether your specific hypothesis was correct. And also you can think back to our plant experiments that we did in weeks 1 through 4, where we put them in different light boxes. So from this graph, we can actually tell that they might do all right in red light, and especially in blue light, but if we put them in green light, would you expect the results to be good or bad? Bad, because it's one of the wavelengths that chlorophyll A and chlorophyll B are actually not very good at absorbing. Luckily, they have like xanthophyll, but this is just a pretty cool way to interpret that experiment we did in weeks 1 through 4. So this is just another way to put this here. Okay, really quickly, for cleanup for exercise 2, just make sure that you are deleting everything you found, so picking out the extra pieces in the big tube and cleaning up all the little scraps of paper around you. Just for the next class, that would be really, really nice. Make sure to re-cork everything because that stuff can evaporate. Okay. So exercise 3, I'm just going to explain it really, really quickly. So people who are already done with exercise 2, move on. So this is where I want you guys to look at organisms whose primary photosynthetic pigment is not chlorophyll. So as you guys can tell with red tides that happen in this area, those are actually photosynthetic organisms, but they're red, which means they're not using chlorophyll as a photosynthetic pigment. So there are other strategies and other wavelengths that different organisms, depending on their environment, will use. So for example, in the deep ocean, one of the first wavelengths to stop is red. Red actually gets absorbed by ocean water in the first few meters. So if you had a photosynthetic pigment that only absorbed red wavelengths, you would not be so hot in the ocean. So this is just another way to think about this, that there are other strategies and other pigments that you can use depending on your environment that different organisms specialize in using. So you're going to have 3 observations you're drawing in your notebooks. You're going to be drawing plankton, red algae, and brown algae. And you guys will use your microscopes you still have out. You can make a wet mount, but all the material you need to make a wet mount should be over there. I'm about to go get the pumpkin out of the refrigerator for you. And let me know if you guys need help making a wet mount or getting an image. Good? Awesome. That's exercise 3. And then lastly, we have exercise 4, just in case you guys can move on. Both exercises can be done at different times. So this is something you guys can actually do on your own and you don't have to come back. So once you're done, you can do this on your own and leave if your station is clean. So step 1 is to find plants on campus and around San Luis Obispo. So this is something you guys can explore if you want to do it. And there's a ton of different kinds of plants all over the place. Because Matt Ritter is really good at trying to do everywhere. But what we want you to do is think about the photosynthetic equation and then look how plants have adapted to different environments and why different adaptations might be favored in different kinds of environments specifically dry and hot environments versus humid and rainy environments versus aquatic environments. So what is available in that equation and what's limited? So if you think about stomata and they're open so they can do gas exchange or whatever, that's super cool when you're not gonna dry out and defecate in the desert. So in a desert, you have an abundance of sunlight, but you have to really deal with water loss. And so that is the way we want you to think about these different environments. And think about those adaptations. Go around, draw them, and even better yet, take photos of them and then write out the adaptations you think they would need for them. And the notebook time associated is table 3.1. So per environment, 1 plant adaptation and why you think that that helps. Okay. Like an extra one. Right, so those are the last 2 activities. And then after that, if your station is clean, you are good to go. If you need help interpreting any graphs or checking to make sure they're labeled, please come by me on this. Yes.